Right. We move on to our next speaker, Martina Canatelli Mallat, sorry. She's doing her PhD from the Biochemistry Research Institute of La Plata, Argentina, with Dr. Rodolfo Goa. And she'll be talking about pattern separation performance and hippocampal neurogenesis in aged female rats. Over to you, Martina.
Thank you, Amita. Hello, everyone. So here I'm showing. Okay. You see that? Well, I'm going to talk about my work, which is about pattern separation performance and hippocampal neurogenesis in aged female rats. So as a brief introduction, we know that aging is associated with a progressive deterioration of learning and memory. Here we have a scheme of the rat hippocampus with its neural circuitry, and we focused our studies particularly on the dentate gyrus and the CA3 region, which are known to be essential for pattern separation. This process consists of the segmentation of similar or overlapping memories into completely distinct memories. So in order to evaluate the effects of aging on the discrimination capacity of Sprague-Dawley rats, we assessed 3 age groups: young, middle-aged, and senile. The rats were submitted to 3 days of habituation and then a sample phase and a choice phase for each one of the behavioral tests. We started with the spontaneous location recognition task. In this task, in the sample phase, rats were exposed to 3 identical objects equally separated between each other. And in the choice phase, one of the objects was removed and one of the remaining objects stayed in a familiar position while the other one was placed in a novel position. So we analyzed the time that rats spent exploring each of the locations. And as you can see here in this graph, the 3 groups explored all of them similarly. For the choice phase, we expected the novel location to be the most explored. And what we found was a markedly diminished discrimination capacity in middle-aged and senile rats compared with the young ones. Then we performed the spontaneous object recognition task. In this task, we have 3 objects in the sample phase, with objects 1 and 2 sharing one feature, and in the choice phase, a novel object was made by combining the 2 non-shared features, and the familiar object was a copy of the 3rd object in the sample phase. We assessed the same parameters as in the SLR, but this time, for the choice phase, we found a statistically significant difference between senile rats and middle-aged rats, and between senile rats and young rats, but not between young and middle-aged rats. Once the tests were concluded, the rats were sacrificed, and we proceeded to study their brains. We assessed immature neurons in the dentate gyrus by doublecortin immunohistochemistry. We also immunostained mature neurons in the CA3 region with Newen marker, and microglial cells in both the dentate gyrus and CA3 region with IVA1 marker. For the doublecortin immunohistochemistry analysis, what we found was a diminution in the population of doublecortin positive cells and also a diminution in the diameter of these cells. So as the dentate gyrus is one of the 2 regions of the adult brain in where neurogenesis takes place, we interpret these results as a decline in neurogenesis in the older rats. For the Newen immunohistochemistry, we also found statistically significant differences between the older groups and the young group, even though we expected to find no differences based on previous reports. Finally, for IVA1 immunohistochemistry, we classified the positive cells into reactive and non-reactive to determine whether there was a neuroinflammation process ongoing. In the dentate gyrus, we didn't find any differences between the age groups, but in the CA3 region, we did find a significant difference between the senile rats and middle-aged rats, and between the senile rats and young rats. And these results are consistent with previous reports that affirm that aged microglia has several impairments, including an increased activation. So as concluding remarks, we can say that there is indeed a loss of discrimination capacity in similar contexts in aged Sprague-Dawley rats, and the impairments arises early where discerning spatial overlapping representations, but not when discerning overlapping representations related to features. We know that the decline in neurogenesis only explains in part what we observed in the spontaneous location recognition task, but not in the spontaneous object recognition task. And we propose that the morphological changes that are taking place in the hippocampus alter the dentate gyrus CF3 connection, leading to pattern separation deficit. On the other hand, the middle-aged rats keep their discrimination capacity of overlapping memories related to object features. Possible explanation for this would be, as Isabel was saying, that there's another pattern separator node which is less affected by age, and this pattern separator node would be the perirhinal cortex. So as future perspectives, we aim at studying this region in order to evaluate age-related changes and confirm this last hypothesis. Thank you for listening.
Thank you so much, Martina. It was really a very interesting